restriction fragment length polymorphism is a backbone of genetic engineering tool. Okay. Not only RFLP, RFLP, RAPD, and AFLP. RFLP, RAPD, AFLP. These three techniques are the backbone of genetic engineering. Why? Because these two techniques are directly find out the homology of the DNA sequences. These three topics are significantly contributed in the CSIR or GATE or other examinations. Just you go through the question paper, previous year question paper. So one or two questions are directly asking from the RFLP, RAPD and AFLP. So more important topics I try to convey. Okay. And also we are already studied the restriction enzymes. So 4 types of enzymes are there. The enzymes are recognize and cleave the double stranded DNA. Is we know all of that information. Okay. So among the three techniques RFLP, RAPD and AFLP, the first technique is called restriction fragment length of polymorphism. So classification of these techniques based on the PCR. So RFLP is a non-PCR based technique. So please keep in mind, it's a non-PCR based technique is RFLP. Remaining two are RAPD and AFLP. Both are PCR based techniques. So this techniques only is the non-PCR based technique. That's why I am learning, and also what are the problems, how we to do the techniques. So we can learn one by one. Before we starting this technique, we should know something. What is called means, what are the times when we are using this technique? It's very important. So just as we go to genetic principles, genetic phenomena, you know very well, you know the phenotype, genotype, these are encoded from allele. So allele means a pair of genes present in the same locus, which encodes the single phenotype. Yes, you know the basic concept of genetics. Apart from this, you know recombination. Yes, you know recombination, and also linkage is very important point. You know linkage, genetic linkage, linkage map, linkage recombination. So number of phenomena is there in genetics. Just to remind the linkage. Linkage means two different genes are present in the chromosome at very close nature. So two different type of genes are linked closer. So if the both genes are the time of replication or the time of recombination, the both genes are simultaneously replicated, simultaneously transformed, simultaneously recombinant into the progeny. For example, you know very well cells are divided every time. Yes. So that time the pair of the different type of genes are linked closer. So the both are simultaneously transferred into the another generation, or F1 generation, F2 generation. So this topic deal with the linkage is very. Similarly, if the two genes are not close means the A gene or B gene. This gene are not linked or present at the chromosome very distant nature. If the two different type of genes are closely linked, so even though the linked genes are transformed or recombinated, they simultaneously transfer into the F1 generation, F2 generation. It's a linkage. Yes. Suppose if the two genes are present in the chromosome at very distant nature, not linked means the individual genes are maybe transferred recombinant at the time of cell cycle. Okay, this is a non-linkage genes. So basically, genes are classified into two. One is linked genes or non-linked genes. I think you know this principle, and I think because you are already studying genetics. Because without genetics, we can never do the genetic engineering. Okay, come to the point. I'm trying to tell where we done the RFLP is the main important. What conditions we are going to do the RFLP and what we are finding, why we are finding. After finding what we concluded is the main important thing. Once again, I'm telling you, restriction, fragment, length, polymorphism is the backbone of genetic engineering. Please keep your mind. Very important technique. They're the basic technique, and also non-PCR basic techniques. Okay, come to the point. If the non-linked genes are easily studied and analyzed by the breeding, the breeding experiment, yes, and also the non-linked genes are produce the visible phenotype. Visible phenotype means for the mental experiment, the mental to take a circle seeds or wrinkled seeds, or red color flower or orange color flower. So this is the visible napped visible phenotype. So this type of phenotype we can easily study it by the breeding experiment. In the case of suppose if the two genes are linked to each other, so at the same time the linked genes are transformed into the F1 generation or F2 generation, it produced not visible phenotype. It not produced the visible phenotype. So that time, how we study the linked genes? So that time we study using RFLP.